Hello, and welcome to Physical and Chemical Properties of Water. I am so happy that so many of you returned to join us for our second session of our webinar series, hosted by 3D Molecular Designs and the MSOE Center for Biomolecular Modeling. And welcome to all of our new participants too. I'm Heather Ryan, Partner and Director of Operations for 3DMD, and I will be your host for the series. Every week before we start the program, and while everyone is getting logged in, I'll share a little bit about our organizations and what we do. 3D Molecular Designs develops, manufactures, and sells molecular models and kits around the world. Our product development team includes educators like you, scientists, engineers, and designers. We employ college students to do production work, which includes operating injection molding machines, dye stamping foam, and packaging components. Our small team is dedicated to providing high quality, effective models and kits at the lowest prices possible. Jim Lane will start us out tonight with the water kit, and we will see how many puns he can work into the session. After a session, Jim will answer as many questions as possible before 7, and then we'll take a quick break before Tim presents aquaporin and the story of exploding frog eggs. After Tim, we will have a Q&A session for both of them, so please stay on and say hello to us as well. I now have the honor of introducing Jim Lane. Jim Lane earned his bachelor's degree in biology and his master's in science teaching. He's been teaching high school biology, engineering, and field ecology in Minnesota for over 11 years. Jim also teaches science methods for pre-service teachers in an adjunct position at Augsburg University. He is both a senior fellow of the Knowles Teacher Initiative and a current member of the HHMI Biointeractive Ambassador Academy. Jim was awarded the Outstanding New Biology Teacher of the Year award by NABT in 2011. He has also mentored MAPS teams for the last 4 years. More recently, he has helped develop and facilitate a teaching fellowship for Tibetan monks who teach science in India and Bhutan through the Science for Monks nonprofit. Jim loves spending time outside with his family, going on adventures, and exploring the wonders of the natural world. With that, Jim will begin with whet your appetite for learning with the water kit. Take it away, Jim.
Well, thank you for the wonderful introduction, Heather, and welcome to all of you. Thank you for coming out tonight. So thank you for those of you who are new joining us, and thank you for those of you who have come to the previous webinar. Tonight, I will be using the water kit, but everything that I will be doing tonight, you will also be able to do with your water student modeling pack. So if you have either of those, the very first thing you're going to need to do is make sure those are in hand in front of you. So as Heather said, my goal tonight is to engage you as students. So I want you to make sure you have your student hat on, but have your teacher thoughts in the back of your head. So I want you, we all have some background with water, but I want you to think as if you were a student and this is new and fresh. So the first thing that I want you to do is I'd like you to, if you have a notebook handy, I would like you to make a notice and wonder chart. We are going to be filling that out as we navigate through the different aspects of the kit. So I'm going to give you just a second to write down a notice wonder chart. So just make a T chart on a piece of paper, and on one column, put notice, on the other column, put wonder. And once you have done that, I would like you to take out the water molecules from your kit. Leave all the other things that also take out just the water molecules. They're red and white. Okay. So the first thing I want you to do is I simply want you to engage with the model. I want you to play with them. I want you to write down as many notices and wonders as you can, just playing with as many water molecules as you have in your kits. So I'm going to take just a short amount of time and let you make some observations and ask some questions. If you, we will be playing a short video that will also illustrate, and hopefully you can make some observations from that. So take just a couple of seconds here, make some observations, write down some questions about your water molecules. Okay, so what I'd like you to do is you should have a wonderful list of notices and wonders. I want you to pick your favorite notice from your list, and I want you to put that in the chat so that we can all see what is your favorite thing that you noticed while you were playing with just the water molecules. So I know it might be hard to pick just one, but let's see if we can narrow it down. Whoa, one molecule can hold up to 30. Okay, red and white, excellent. A fluid clump. White only sticks to red. All right, very good. Okay. There's some very astute observations here, and there's some very basic observations, and that is fantastic. Okay. All right. So these are notices. And again, I want you to think as if you're a student, these are the kinds of observations that we want our students to make. Even though they might be simple, we have to get excited about it and acknowledge that. So as simple as that they're red and white, that might be something that they notice, but that's an important property of water, that we have this 2-to-1 ratio of hydrogen to oxygen. As simple as that is, something that as teachers we might take for granted, that's a way we can engage with students. I can look at is the attractions. I saw the attractions a lot, that the white and the red are attracting and the white and white are repelling each other, or the red and the red are repelling each other. So again, another very important observation. Also notice how I'm not going right in and saying oxygen and hydrogen. I can start off with just saying white and red so that we're building these observations, honoring these observations as to further engage the students. Ah, yeah, so Brian just put in different sizes of red and white so that the red oxygen is huge compared to these kind of small little hydrogens that are attached on there. So these are fantastic. Higher level observations might include that there's an angle. It's not a straight molecule. There's an angle, the kind of the oxygen on one end and the hydrogens are on another end. And I'm kind of looking through the chat, and yeah, the bond angles that you can make rings, that you can do all these things. So these are all fantastic. Hopefully try to make sense of these. I will again repeat that I want this to be accessible for anywhere from middle school, elementary even, all the way up into college level. So as we progress, I want you to think in the back of your head, how might this work in your context? And how might your students engage differently based on their prior knowledge and learning. The next, first off, I think there's no questions. I think we've answered those. I don't know how to clear the questions. I'll work on that. Next type. So again, keep the water molecules out. And I want to talk about the different bond types and how we can model that. So what I'd like you to do is I'd also like you to pull out the blue and the green spheres from the kit if you have them, and have those at the ready. We have three different types of bond types that we can model with these. The first one is covalent, and that is the kind of bond that we see between the hydrogen and the oxygen here. And what I want you to do right now is I want you to take just your fingers, no fingernails, just your fingers. I want you to pull the hydrogen off the oxygen. So with your fingers, just your fingers, no fingernails, pull it. I can almost hear some of you like trying. Try a little harder. You just can't do it. You can't do it. And that is the glory of the covalent bond. It is a very strong bond, that there is that shared electron between the hydrogen and the oxygen, and it is a very strong bond. Comparatively, taking two water molecules, what you'll notice is that there's that attraction between the red and the white. And when we see that, that's a much weaker bond. That's a very weak bond, in fact. So that observation right there, that's an important observation to make. The difference in bond strengths tells us something about how these molecules are going to interact and behave with each other. And finally, we have these two. This is sodium chloride. So we have our little sodium here and our big old chloride in the green. And we can pull those apart and we can feel the difference. So we can physically feel the bond is a little weaker than the ionic bond. So here we have that complete transfer of an electron. And what we can see is that physically we are engaging with the model. And we can now not just say, oh, it's a shared electron, it's a transfer of electron, it's just an attraction. We can feel it. So what the most awesome part is this is now an experience. The students can experience the strength of those bonds in real time and really get an idea for how relate to each other and the importance of that. I am going to get maybe not to that answer, Patricia, but we will maybe try to address that in our next section here. Why this is important, talking about covalent and hydrogen bonding. This is something that I do in the very beginning of the year with my sophomore biology classes, and I come back to it throughout the semester, throughout the year. When I talk about DNA replication, or if I'm using the Flow of Genetic Information kit, this is why the DNA is between the helices rather than across, because the backbone is covalent bonds, those phosphodiester bonds, but the attraction between the nitrogenous bases is a hydrogen bond, a very weak bond. So as simple as that is with the water kit, it's a concept that we use throughout biology to hopefully make sense of the molecular world. Will this cause students to think ionic bonds are weaker than covalent? There is a differential in charge, perhaps, yes. So we see that the ionic bonds are slightly weaker than the covalent bonds, for sure. So that might be something that you can address, but overall, the idea that they can experience the differential between those bonds, I think, is something that is important. Are the sizes of the hydrogen, sodium, chlorine, etc., to scale? As far as I know, I see Heather is typing. And then finally, could you please show the sodium and chlorine model again? It isn't sodium bigger than chlorine. So we have sodium and chlorine here, sodium in blue, chlorine in green.
Hi, Jim.
Okay. So
they are relative to size, and Tim can always speak on that more at the end of the session.
Great. Thank you, Heather. Did I switch them? I don't think so. The next thing what we're going to do is we are actually going to model the hydration of sodium chloride. So what we have is I would like you to pull out a sodium chloride. Now this is just two little ions coming together. And at the very end, I will have Heather. I think it's going to be really cool. So the first thing that I want you to do is take this, and now you're going to need all the water from either your student modeling kit or the actual full water kit. And I need to have all that out on the table. And what I'd like you to do is one by one, I want you to start adding waters to the outside of this sodium chloride. Okay, so as you do that, I want you to take some, again, notice and wonder, like what here. And what I'm hoping that you'll see is that we end up with, and I don't know if you can see this as well, but right in the middle, we have this little tiny sodium in there, and we have it completely surrounded by water. But it's just the oxygens that are attracted. And here we have the same with the chlorine, but it's just the hydrogens that are attracted. Okay, so what I want you to make an observation, or what I want you to think about here, is that when we have chlorine in solution, it's completely hydrated. And when we say hydrated, what we mean is that it is completely it is covered with as many waters as it can possibly carry. And likewise with sodium. And so what we see here then is hydrated sodium and chlorine, that those ionic bonds that were previously there can no longer get together. And if we can imagine this in a cup of water, what we'll have is water that is in between all of these different ions, preventing them from coming together. So when we talk about solution or a solute, we can now think about that, and we can see, why is it that when I stir salt into water, why does it disappear? Where does it go? It disappears because they are hydrated. I believe there's a next slide where I illustrate boiling of water. I wonder if we could get that up there. So I will model this. I'll model this outright. And so what I'd like you to do is, I want you to imagine this as in solution. So we have the solute, which is the sodium chloride, so sodium and the chlorine. And then we have the solution, or the solute, which is water. Now, what happens if we boil that water? Well, what we're doing when we boil water is we're providing energy to each of these water molecules. And these water molecules then get enough energy, they disappear, they turn into the gaseous form. So what we see then is that now some of these chlorines, or I'm going to give some energy to that sodium there, and we have that some of these chlorines and sodiums stop becoming hydrated, and lo and behold, what starts forming is sodium chloride. So what we'll see is that from that solution, what was previous in solution, as the water gains energy and goes away, we can now see that that sodium chloride will be left over. So we can talk about the idea of we can look at water in the liquid form and how much energy it has. We can talk about the amount of energy that it would take to make this occur, but then we can also see and make sense of how a solution can be really, how a solute could be precipitated out of solution by simply removing some of the water. Let's see. And with that, the other thing that I want to touch on here is this would be great for when we look at states of matter. So I'm going to try different rings and just throw together a ring here. So if we come together with a ring, this could be a solid, and we can see that there's a hole right through the middle. That can help us explain why water might expand while when it goes from liquid to solid state. You can also have students model the liquid state by spreading the individual water molecules out on the table and explaining what that would look like. And then ultimately the gaseous state. Now there's water molecules everywhere. So this is a really dynamic. It's accessible at different levels, but also allows you to model a lot of different ideas. And then finally, you can talk about cohesion and adhesion, which you can really see, like, obviously cohesion, we can see that water is attaching to other waters. There was the observation early on that one of these water molecules can hold 13 other water molecules. So we can make sense of some of these observations, like why do drops of water on leaves, why do they have that huge, like almost perfect sphere? And that is because of the cohesive properties of water. Is adhesion, where we have water sticking to, well, in this case, sodium chloride, but how does it stick to other surfaces? And we can start thinking about that in terms of hydrogen bonding or other attractive forces that are occurring. And now I'm going to introduce Heather. She is going to model supersaturation. So what we have here, if you look in the center of the screen, there is a large sodium chloride crystal. So that's like this, only larger. And if we know salt crystals are turned, and what we can see is that there's already some hydrated chlorines and hydrated sodiums around the outside. And what Heather is going to model is a supersaturated solution. So what that means is there's not enough water to hydrate all of the chlorine and all of the sodium. So now what you can see is that she's running out of water molecules and there's still some sodium chloride left. So this is a great opportunity if you have a class set of these sodium chlorides together, and you could model this and have them explain why is it that if I pour sugar or if I pour salt into water, that there's a certain point where the salt stops disappearing, that there's still salt left on the bottom. It's because we have become supersaturated, which simply means that there's not enough water to hydrate all of the sodium and chlorine that are currently in solution. Now I will, okay. There's no access to the question, but I want to make sure I provide opportunities for those questions. So if you have a question, this would be a good time. I hope that your appetites are being whet. Okay, so if there are not questions here, what I want you to do, oh, yes. Okay, so Gabrielle, yeah, you could do diffusion, and that's one thing I forgot to mention. You could measure, you could even model concentration gradients of water, or with the sodium and chlorine ions, with them hydrated, you could model concentration gradients and explain why things are going to move certain ways, and really get to the idea of diffusion by doing so. So yeah, that's a great, and you can a great question. You can also look at the differential in size between the hydrated sodium and the hydrated chlorine, that they have a different size, and use that to illustrate rates of diffusion and why different sizes are rates. I would like to know what you did with the sodium and chlorine as like right now. I just hit it. I took it away. But this is where you could, if I was in class, I'd be much more animated because I would not be limited to the small screen. I would be making this thing, giving it lots of energy and flying it up and showing it go into the atmosphere as it's happening. That's how I would model that in class. So yeah, again, this is something where I would be not limited by the constraints of my box. What I want you to think about right now is I want you to kind of put your teacher hat back on and think about how does this model change how you might model or teach solutions and states of matter. So how could this modify how you currently think about or teach solutions, states of matter, standard biology, chemistry terms. You don't necessarily need to put this in the chat. If you have something that you just have to share, please do. But think about this. Think about how we currently or maybe more traditionally might teach things like solute solution, and how this might change that as an opportunity to engage students with their learning. And then I want you to think about how this model might help support because that is the next step. The last portion here, I'm going to dig into the polarity and then talk about other kits. So yeah, Jennifer, you're asking a question about to model how glucose or any polar covalent compound. I think I'm going to address that question in the next session when I talk about polar compounds and nonpolar and amphipathic. Heck yes, Mary, I agree. That if you can make it, it's a good day for everybody. All right, coming back to our water kits. What we are going to do is we are going to talk about polarity. So one of the very first observations that we made was the idea that it's not a linear molecule. In fact, there's this offset of the hydrogens compared to the oxygen. And that offset is what makes water the most gorgeous and wonderful molecule that allows life. So because there is that offset and this oxygen, I always ask my students, like, what's the one thing you learned in kindergarten? They're like, sharing is caring. I'm like, oxygen didn't go to kindergarten because oxygen does not like to share as well as hydrogen. And so what that means is that we have an unequal distribution of a probability of where an electron will be, favorable towards the oxygen and away from the hydrogens. That distribution of charge is that polar property of water, that we have a slight positive charge favoring the hydrogen side, and we have the slight negative charge favoring the oxygen side. And so that very simple idea of a polar compound is going to be essential for getting into talking about things like glucose, talking about membrane transport, and talking about macromolecules and protein structures. So this one single model is the gateway to the rest of the molecular world. And so what I'd like you to do is, if you have the student modeling kit, you can take out the little tiny methane. And what I want you to do is I want you to look for there's one of the hydrogens that it's a very special hydrogen. It has 3 little arrows around it. And that one you can pull off and you'll see something like that. If you have the student kit, you'll have ethane. You'll have ethane, and I want you to look for the same, there's one hydrogen that has 3. And we are going to do that. Oh, I just got ahead of myself. I want you to not do that. I want you to take one step back, and I want you to take out just the ethane or just the methane. So got a little excited about polarity. I know it's exciting, so I have to slow my roll. Oh, Tina. Oh my gosh, Tina, yes. Holy cow. One of my this is, after we model, what does this model do well? What are the limitations of this model? Anytime I talk about models or at any level, that is what we have to do. As science-minded people, we have to be able to evaluate the models and talk about, all models are wrong, but some are useful. So how was this model useful? How was it not useful? When might it be useful or not useful? All right. Whew, man. So let's think about methane. Can you guys smell what I'm cooking? It's methane. Gross. What I would like you to do is, I want you to take the cup if you have the cup. If you have the bag, I want you to take the bag, and I want you to take ethane, or I want you to take methane, and I want you to put it in the bag or the cup. So I'm going to use the cup. I have the ethane. I'm going to drop the ethane in. I'd also like you to drop in all of your water molecules. So one literally just flew across the room. That is subconscious on me. What I would like you to do now is I want you to shake this thing. I want you to mechanically agitate it with joy. So you're mechanically agitating and you're shaking it and you're shaking it. I want you to mix that thing up. I want you to just let those things go. Get some energy into the system and make some things happen. And then what we will see is the ethane isn't interacting with, maybe I didn't shake it enough. Maybe that's what it is. I better really have some energy this time. Okay. I'm sure that sounds great on Zoom. It's just this horrid sound of things rattling. All right, and lo and behold, even though I provided even more energy, this ethane is not matching up with these waters, but this doesn't make sense. This doesn't make sense now. So we need to make some observations and try to make sense of it. So when I talk about this with my students, I have them look at the ethane molecule and I have them look at the water molecule. And we had previously talked about how this ends. Now if we look at ethane, we have a very symmetrical molecule. We have the 2 carbons and we have the 6 hydrogens. So we're making basic observations about the molecular structures, but we're also being able to notice that they're behaving differently. I will often have students like, well, this one just doesn't have the magnets, Mr. Lane. Like, yes, but why? Why in that model, when we model that, why wouldn't we have magnets here, but have the magnets in water? So here's another opportunity to access and the high level chemistry is talking about the physical properties of each model. That we have a symmetrical molecule here that has two elements that are equally electronegative. So they share well. Ethane went to kindergarten. They know how to share with each other. Hydrogen, carbon, they get along. And then we have that oxygen that just does not share well with each other. And so we have that attraction between other molecules. And so this is where you introduce the idea that it's not able to bond with the polar molecules because of that unequal sharing of electrons, but also because of its chemical makeup. That ultimately it's because it has different elements in a different conformation that gives them that property. So what I will do now we can talk about the arrows. This is exciting now. So now I want you to take your fingernail and I want you to pop off. I want you to see, you see the koala bear. Yes, we see that. I want you to make a koala bear. It's adorable. If you have methane, you're going to make just a koala bear head, which is slightly disturbing, but equally cool. And then in your modeling kit, you will find a simple hydroxyl group that is a water without an oxygen. And I want you to combine that oxygen, that hydroxyl group, to make our favorite molecule, ethanol, or methanol if you only have the methane. What I can do is now I want to redo it. First off, this is with the students. You have to acknowledge that this looks like a dog. Like it's a little dog. It's an adorable little ethanol dog, and it's got a little tail, and the kids love it. Just trust me on that one. So what I would like you to do now is I want you to do the same thing we did before, taking the cup with the bag, and this time I want you to put ethanol in. So ethanol in. Don't try to drink this. I know it's a weeknight, it's a late night, but do not try to drink this. And shake it up again. So now what we will notice, and you can see very quickly, an attraction now. I can do methane as well. Yes. So here we have that ethane, and if this was methanol, it would be exactly the same. I will do that right quick here. Oh, where's my other hydroxyl? There's one. So if we have the methane, I'm going to pull off the one, and I have just my little koala bear head, and I'm going to put that on there, and there is methanol. So there is methanol. So now what I can see is that I have that ethanol and I have methanol. Both of them are attracted now to that water. So previously, when it was just ethane or just methane, we did not have that attraction, but now we seem to have that attraction when we put on that methyl group. So what we can take, and again, I'll use ethanol here. You can take that water molecule and you can see that we have that same attraction that we had with other water molecules found now on one side. Whereas the other side, if we look at the tail end of the dog of ethanol, we still don't have that connection. We don't have that attraction. And that right there is what we would call an amphipathic molecule, that we have some portions of it that have a polar property and some portions of it that have a non-polar property. And that, like this molecule, is why proteins fold the way they do. When you have those hydrophobic and hydrophilic regions that are folding and causing different bends and turns, that's what we are modeling right here. So you can introduce not just polarity, but also hydrophobic, hydrophilic. This is where we can start talking about membranes. We can talk about, well, why is it that we have a polar, non-polar region? Because, well, we have all of these oxygens. And this is where you can have students start taking spaces and looking for those oxygens, looking for the nitrogen. So they're like, oh, there's the polar region. There's going to be some hydrogen bonding going on there. There's going to be something different is going to be happening there as compared to a non-polar region. So in all of its glory, this one kit, we are now really accessing opportunities to talk about some very, very cool biochemistry. So I think that is, I do want to, I think, Chris, if you could pull up. Yes. So here is an amazing resource produced from 3DMD, where it is an interactive way where you could assess this. So we are distance learning now. So whether you have these models or not, this is a way that you could assess and have kids illustrate and then annotate and explain why things are occurring. So they have these kits where you can see Chris is manipulating it right now, and then what's happening with their models. I see this as an excellent opportunity for a formative assessment, for students to reflect and actually think about what they were doing and to annotate so that you kind of know what they're thinking and illustrating their observations. And then finally here, as I've been saying throughout, this singular kit is the gateway for so many other kits. I use this with the Flow Genetic Information Kit, which you see on the right. I've used it with the membrane kit when we talked about sodium moving hydrated through the membrane rather than potassium moves, it dehydrates itself, it goes through the channel, then gets picked back up. We can use it with the lattice of sodium and chlorine. And the amino acid starter kit, this is how the amino acid starter kit is laid out, is with polar and non-polar hydrophilic and hydrophobic. Right there, it gets you into protein structure. So this is the very first thing I do with my MAPS teams every single year, is we get to know the water kit in and out, because that's what's going to help us make sense of all of the other things that we're going to explore. And with that, I see that we do have a couple of questions. Oh, I answered the one about models, I think. And I did see something in the chat about having trouble with the, And that is a very normal thing to do. And I just simply do not have enough focal length on my camera here. But there is one hydrogen that has 3 tiny little bumps on it. They look like little arrows pointing at that hydrogen. That's the one that you should take off. So that is the one I just simply cannot get in close enough. So thank you for playing along with me tonight. We do have a couple of extra minutes here that if you have questions or like me to mention, or I think Chris and Heather could put microphone privileges on.
Great. Thank you, Jim. Wonderful presentation. There, we do have Tina's question about limitations of models. And do you want to talk for a few minutes about common things that you discuss with your students about limitations of the WaterKit model? You know, what it does well and what it doesn't do well? Oh, you're muted again, Jim.
I just lost my headphones. Could you repeat that for me?
No problem. So Tina was asking about the limitations of models with the WaterKit. And so is there something that your students typically show or talk about in terms of, you know, what the WaterKit doesn't show well?
What the WaterKit doesn't show well? Well, I think that like electron dot structures, it doesn't show the actual things that are happening there. It illustrates how those structures within the molecule are behaving and interacting with each other, but it doesn't illustrate that relationship as well in its own right. So the idea of a shared electron versus a transferred electron, that's a limitation of this model. It doesn't, not something we can explain, but it's something that we can see the effects of. So that's a perfect example I would use.
Great, thanks, Jim. And I'm going to try to do my overhead camera and show you guys. We'll see if we can get close enough to see here. So you can barely see this little dot there, and there are 3 of them as you go around. And that's the one that you take off the hydrogen, and it could be that you will still have a post showing up in there, and you can use your nail or a pin to just snap that out. And if you take off some of the others, you will see that there's a post there that you definitely can't take out. So, you know, if nothing else, by trial and error, you can figure out the correct one to take off. And then you take one of your hydroxyls to just put on there. And I can't believe I've been using these models for, I don't know, 15, 20 years, and I never saw that koala face before, Jim. So that's something I'm going to talk about all the time now. So let's see.
And molecular movement.
Come up.
So I want to address the idea of molecular motion, and that when we have a molecule in gaseous or liquid form, it has more energy. So if we imagine that it's flying in and it hits this other water molecule, if it has enough energy, that the force of that hydrogen bonding is just not going to be enough for them to interact. It's going to hit and bounce away. So you can really illustrate like how fast can you have it go where it's actually going to stick. And that is really modeling the amount of energy and the difference between a liquid, a solid, and again, they have that hydrogen bonding and they're still wiggling. So I don't know if that's what you're asking, Paul, but that's another thing you can look at.
Okay, what else? I did write a couple of other questions down as we were going. And so there's one question about the ring with a ring of 5 or 6. I'm going to go back to my overhead camera. And I know that like when you just play with the water molecules, well, apparently we are having a lot of technology problems tonight. So I thank you all for bearing with us. But maybe I am frozen now.
Mhm.
So go ahead and
We-
take it away, Jim.
We can hear you, Heather. We just can't see you now.
Jim, if there's another question or something that you want to talk about, go ahead.
Yeah, are there other questions? Is there ones that I have missed in the chat or missed in the questions? If you
Jim?
could put, yes.
Can you address the hydroxyl group and removing the hydrogen cap from the water molecule? I will just say that one of the problems, so someone, maybe multiple people are having it, but from the actual water instead of the methane, is that those caps can be a really tight fit when you first receive them. Can you show that, Jim?
Yeah, so you don't ever remove, and this is something that I will, if you're a classroom teacher, do not ever remove the hydrogens from the oxygens that are in full waters. You are only going to want to find that hydroxyl. There is one oxygen with that's missing a hydrogen in the kit. Please use that because what kids will do, and the students being students, is they will take from the ethane or methane, and then they will put it on a water, and then you'll realize that you have a hydrogen on a water that doesn't have a magnet and isn't behaving. So you're always going to use just that hydroxyl group, and then to find that single hydrogen in ethane or methane, you're going to look for these just 3 tiny, tiny little bumps right around the ring, the plane where the 2 atoms will connect.
Phyllis, I believe that the student
The
might be
hydroxyl in it.
supposed to have a hydroxyl group, so it could be that there was an error in packaging. If you don't have the hydroxyl group, maybe you could just send us an email. And I think someone else had mentioned that they hadn't received their student modeling kit. And if you can let us know when you've registered, if you didn't receive the kits, that to just shoot us an email and we'll work that out as well.
Acid base lesson. I have not used this model to teach an acid base lesson, and I don't want to punt on that one. But I certainly can see the benefit of doing that and the variability or the versatility of this model in teaching acid base. I just have simply not used it to do so.
Tim, I'm going to let Tim come on and talk about a couple of things. And what I was going to say, the post office is taking an extremely long time to get packages shipped or get them out to people. So it's taking about a week longer than we would expect. So and if you got the email with the tracking number, things are on their way. And Tim was going to come on and was going to talk about this, and maybe he can do some of the other things.
Well, I don't know. You guys have asked some tough questions. I'm glad I don't have to tell you why water seems to form 5-membered rings more easily than it does 6-membered rings. Now, if someone knows the answer to that, they should let me know.
I have the model if I could show you guys.
What? You have an answer to that question?
I do. So, but I'll do that at the end.
Yeah, well, I just want to do, I want to show you something I always like to do. Jim has shown you a methanol, right? And then he showed you an ethanol, and both of those small alcohols are soluble in water. They interact with water, so you saw that. But now you can push this one step further and you can begin to worry your kids a little bit, because you can just put a whole string of carbons onto this hydroxyl group. So we can go from methanol to ethanol to propanol to butanol to pentanol to hexanol, et cetera, et cetera, et cetera. And the point is, this is still an amphipathic molecule, as Jim said. But how does the kid know? Let's go back even to ethanol. You know, a student might say, wait a minute, there are 2 hydrophobic carbons here and only 1 hydrophilic hydroxyl group. So I don't think this should dissolve in water. Okay, well, we know that's not true because it does. We've done that experiment. But in fact, as you continue to add more hydrophobic carbons onto a primary alcohol, you will very soon reach a point, and I think it's either at heptanol or for sure octanol. An 8-carbon chain with 1 hydroxyl group is not soluble in water, which I guess I'm supposed to be looking straight ahead, right? So this is a fun thing to do. Octanol is a colorless liquid that looks just like water. But if you fill a glass bottle half full of water and then you fill the other half full of octanol, you can shake that up real hard and you can set it down. And within 15 seconds, the two liquids will separate and there will be an absolute clear boundary between the octanol on top and the water on the bottom. So in fact, this is how chemistry, this is why chemistry gets hard for kids, because you have to develop almost an intuition to know that even though this amphipathic molecule has a polar head on it, at some point the hydrophobic tail on it overwhelms that polar property here with a single head. So chemistry can in fact get complicated as you start adding more and more atoms together in very complex combinations. All right, that's, I don't know, do you want me to say anything more about this? Or, Jim's done a wonderful job of talking about how you introduce these basic principles of chemistry to kids with the water kit. You can do acid base. If you take a water molecule and you take a proton off, at a little higher level of chemistry, there's something called a hydronium ion, in which you have 3 hydrogens associated with a water. So that's a plus charged hydronium ion. The other thing you can do is you can make, this is sodium. So this is sodium hydroxide. That's a base. Then you can take another water molecule and break it, and you can make this, the chloride. This is hydrochloric acid. And you have to be careful about this because I think as chemistry teachers, you go to great lengths to tell kids never pour acid into base. Bad things happen when you do that. But in fact, if you have sodium chloride and you have, sodium chloride. What is sodium?
Hydrochloric acid.
Hydrochloric acid.
Yep.
Yeah. So you can form water from this. These hydrogens come back and you form the salt, then the sodium chloride and water. So that's a very, very simple acid-base demonstration. Not done very well, but I haven't tried to teach this to high school kids.
Okay, what I was going to try to show is if you take a ring of 5 water molecules, which is what is actually, which commonly happens when you just have the water molecules around. But if you look really closely, you will see that there is a little gap in between some of the bonds, that they aren't fully formed there. And if you do the same thing then and create a ring of 6, let's see if it's not in front of me. And if you really look at all those connections, you will see that those are firmly seated on all of them. And I will teach you how to make this ring of 6 at the end if I can get the overhead working again. But we do have our instructions online. And so when you compare that, this is actually much more stable. Where's my ring of 5? And so even if you take both of these and shake them about the same amount, you'll feel that the ring of 5 is not as strong as that ring of 6. So that's something that you guys can test yourselves. Okay, thank you. We are back now. And so thank you, Jim, for that session. And I hope that you guys can join us for a summer course and can spend some time really digging into these models and working with us. And we'll have lots of time for this discussion where we can all be on screen together or in person together. So to introduce Tim Herman is the founder and director of the MSOE Center for Biomolecular Modeling and the founding partner of 3D Molecular Designs. In addition to grant writing, model development, and digging into recently published research papers, he enjoys spending time with his 4 grandchildren. Tim and Diane had the water molecules out this past weekend with the grandkids, and they were modeling ice. And so here are some pictures they took. I'm looking forward to when he starts telling them this aquaporin story. And now here's Tim.
I think it'll be a while before the grandkids learn about aquaporin. All right, so that was good, Jim. So now I get to follow that with the fun part, which is telling stories about proteins. So the protein we're going to focus on tonight is aquaporin, the water channel. So the story of this protein is an amazingly recent story. And by that, I mean that it was not until about 1992, which was like yesterday, that we actually discovered this protein. So leading up to 1992, people knew that the transport of water across a membrane was a very important thing. And they knew that in some tissues, that water transport happened much faster than could be explained just by the simple diffusion through a lipid membrane like this. This is just a small section of a membrane, phospholipid leaflet on top and on the bottom. And if you take a water molecule, it doesn't just easily move through. So this is an impermeable membrane to water. And again, people knew that in some tissues, like your kidneys, there's a lot of water transport that moves back and forth across membranes. But it wasn't until 1992 that a guy named Peter Agre discovered the water channel. And that work was recognized in 2003 with a Nobel Prize in chemistry. And now I don't know, do we have that slide up?
I think that was up,
Okay.
but I'll go ahead and put it up again.
All right, so the 2003 Nobel Prize in Chemistry was shared by 2 people, Peter Agre for his discovery of this protein, the water channel aquaporin, and Rod McKinnon, who had solved the structure for another channel, a potassium channel. So we'll be actually talking about Rod McKinnon's potassium channel in a later webinar in this series. But for right now, let's talk about aquaporin. And I'm just going to do a quick story here. And know that there is an 18-minute video. It's supposed to be 10 minutes. But the guy who did it just talked on and on and on. And it turned out to be an 18-minute video that tells his story. So if I go too fast, if I'm incomplete here in what I say tonight, there'll be a video posted in the next day or two in which all of this is probably said in a slightly more organized way. If we can have this slide up, this is a screenshot of a paper that appeared in 1992 in Science magazine. You can see that the senior author there is Peter Agre. And let me just read the title. It says, appearance of water channels in Xenopus oocytes, those are frog eggs, expressing the red cell CYP28 protein. So the appearance of water channels. So this was the classic paper, which ultimately won Peter Agre a Nobel Prize several years later. But this is when they first were able to demonstrate that a protein that this group had identified as being an abundant protein in red blood cell membranes was in fact the water channel that everyone sort of knew must exist, but no one knew what it was or how it worked or what its structure was. So this is an interesting paper, I think, to look at. It's an interesting paper to have your kids look at because it really gets to the process of science. How do important discoveries like this get made? And let me just say it as an aside, Peter Augrey is a very interesting guy. And there will be some resources at the bottom of this video that you can go to learn more about Peter. There's an interview with Peter Augrey on the so-called MAPS team website where we have an aquaporin module. And it's wonderful to listen to Peter himself tell you about his life, his very interesting high school experiences, where he got a D in chemistry. So that when he got the call, in October of 2003, early in the morning, he gets a call from Sweden, someone telling him he's won the Nobel Prize in chemistry. And the first thought that pops into his mind is, chemistry? What the hell do I know about chemistry? He got a D in chemistry in high school. But his father was a chemistry professor. So maybe that made up for it. Maybe that's why he got a D, I don't know. But anyway, you really need to read something about Peter. It's really a very interesting story. All right, but that's not what I want to tell you about. Peter and his group, Peter is an MD, okay? And he was interested in the Rh factor. He was trying to find the protein in red blood cells that was in fact responsible for this Rh phenotype. And lots of people had looked for it, no one had found it, and he didn't find it either. But along the way, when that project was faltering, they were isolating proteins from red blood cells, and there was this one protein, about 28 Daltons. I guess that's why it's CHIP28. It's a channel-like protein, 28 kilodaltons. So they found this protein that was easy to work with. They could isolate lots of it. It formed a nice band on an SDS gel. So they thought, why not? Let's pursue this a little further. And slowly, as they started thinking more about this protein from red blood cell membranes, where there is apparently rapid transport of water across the membranes. I certainly wasn't aware of that, and I don't know that it makes much sense to kids that you would naturally expect that there'd be a lot of water transport in a red blood cell, but let's just assume that was known by clinicians like Peter at that time. So they pursued this. Just the question was, what is this protein? What's the function of this CHIP28 protein that they had isolated and were beginning to study? So if I wanted to make this story short, I would just go to this next slide, which shows a figure from that 1992 paper. And it says that they knew the amino acid sequence of this unknown protein. And I really need to say something about how they knew the amino acid sequence of an unknown protein. This is really what I think you should engage your kids with. So, we do a lot of imagine that. Imagine that you were a student in Peter Augrey's lab back in 1990, and you were looking for a project to do in Peter's lab, and the real main work of the group wasn't having much success, but Peter kind of threw this project at you. Here's a protein, it's easy to isolate. Why don't you figure out what this protein does? So what would you do at that point? And again, we don't have time to get into this. You need to come to a summer course where we can really have extended conversations. But suffice it to say that if you purified a protein, which they had, you could then begin chopping off the amino acid sequences from the N-terminal end. And you can't sequence the whole protein. It's turned out to be a 269 amino acid protein. But you can determine the first 3 or 4 or 5 or 6 amino acids. Now, if you know the sequence of the first 5 or 6 amino acids, you can get out your table of genetic codes, and you can then write down the nucleotide sequence that would encode that 6 amino acid sequence. So now you've got a probe, you've got a piece of DNA which is complementary to the beginning of the gene that encodes your unknown protein. So using that little probe, you can then clone the gene for CHIP28. All right, so let's just assume they've done that. They've got the gene, and of course, by 1992, we were sequencing DNA. So you then sequence the gene, and you get out your table of codons again, and you use a computer algorithm that just translates that nucleotide sequence into an amino acid sequence. And that's how they knew that it was a 269 amino acid protein. And then they looked a little closer at the sequence, and they produced this so-called model of aquaporin back in 1992. So they looked at the sequence and they said, it looks like there are 6 membrane-bound, membrane-embedded alpha helices. And there they are. And then, as you know, they weren't biochemists, but they had some people who helped them. And they could determine that the N-terminal end was inside the cell. So those alpha helices, if they're sitting in a membrane, then everything below those alpha helices is inside the cell. Everything above is outside the cell. So both the N-terminal and C-terminal end were inside intracellular ends inside. And then there were loops that connected the various 6 alpha helices. All right, and then you might say, well, how did they know they were membrane embedded? Well, you know, if this is going to sit in a membrane, okay, here's the membrane. This is all hydrophobic, right? So these have to be hydrophobic side chains all along these alpha helices. So they looked at this and they saw all hydrophobic alpha helices. If this were a soluble protein, you'd expect lots of polar side chains. So the fact that they were all hydrophobic side chains here said, it's buried in a membrane. So this was beginning to build a case for this thing might be sitting in a membrane. It might be somehow transporting water in and out of cells. So then they did the famous exploding frog egg experiment. And again, this is all about the collaborative nature of science, because Peter's group didn't know how to micro-inject mRNA into frog eggs, to Xenopus oocytes. But someone else on their campus did. So they went over, I don't know if they went over there or someone visited their lab, but they got together. And a postdoc in the lab at that time, Greg Preston, was lucky enough to be the person who did this experiment. And it's such a simple experiment. You take the messenger RNA for this unknown protein, and under a dissecting scope, a frog egg is about 1 millimeter in diameter. So they're huge by cellular standards. So you put these frog eggs in a little petri dish under a dissecting scope, and you come in with a micro manipulator, sharp needle, and you actually poke a hole, you impale one of these frog eggs, and then you inject a little bit of liquid. And of course, the liquid in this case contains messenger RNA for CHIP28. And then you pull the needle out, and lo and behold, you haven't done drastic damage to the frog egg. And it'll sit there in this isotonic solution, and it'll be just fine. So he injected a series of these frog eggs with just buffer or with the mRNA for CHIP28. And then one by one, he would take these frog eggs and he would drop them into another petri dish that had just water in it, not isotonic buffer, but water. Okay, and normally a frog egg lives in water, right? So normal frog eggs won't do a darn thing. They'll just kind of sit there and be very happy. But what they found is that over a very, very short period of time, up to 3 to 5 minutes, you would start to see these frog eggs that had been injected with the mRNA for this protein. They began to swell up. And they got bigger and bigger and bigger and bigger, and then they started to pop. So Greg talks about the time in the lab when they were just pop, 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 pop. He'd take a dozen of these microinjected eggs, put them in water, and then at about the same time, between 3 and 5 minutes, he would see pop here, pop, 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 pop. What an exciting experiment, because Usually it takes hours and hours to get a result. And it's not nearly as impressive as frog eggs popping right there as you're watching. So that was the classic experiment, and more than anything, that is probably what won Peter the Nobel Prize in chemistry. All right, and there's a chart there that shows some data. I know that one of the things I think we need to do for high school kids is give them practice interpreting data of this sort. This is a very simple graph that just shows the swelling of the injected frog eggs over time. And you see that little X up there at the top, that's when the egg popped. All right, so that's the story, but what about the structure then? So at this time, can they see that yet?
They can, but we'll come back later.
All right, I want to show you the model. So I'm going to hold a model here in front of you, and let me see if I can explain what's going on here. At that time, people were beginning to discover and solving the structure of what was known as 7 helix transmembrane proteins, or 7 transmembrane helix proteins. I can never remember exactly how you're supposed to say that. But they were proteins that consisted of a bundle of 7 alpha helices, and these spanned a membrane. So again, if this is a chunk of membrane, bring up here, you can see that it's just large enough that it would go from one side of the membrane to the other. So they began finding all sorts of 7 transmembrane helices. And you remember that model I showed you a little earlier, back in 1992, it had 6 alpha helices. All right, so let's, this is the structure of the aquaporin, which was solved several years after 1992. Peter again collaborated with another group, structural biologists, who actually crystallized this protein and solved its structure. And what they found is that there were 6 transmembrane alpha helices. So let me count them for you right here. I hope you can see this. We're going to start with this green one. So there's 1, 2, 3, 4, 5, 6, 7. And the key, of course, is the 7th alpha helix, which Peter didn't know anything about. If you look very carefully at this 7th alpha helix, you will see that it's not a continuous helix. It starts out down here being orange, and it's actually a half helix that goes halfway through, and then that orange backbone turns around and goes back out here. At the same time, there's a green half helix that comes down, butts right up next to the end of the orange helix, and it turns around and comes out. So this is, in essence, a 6 transmembrane helix, a 6 plus 2 halves, which is 7. So it's a bundle of alpha helices that sits in a membrane. It's got a channel in the middle. And let me pull off a couple of the helices, and it reveals another thing. And let me stop and say something about using models like this. You know, we're struggling here to actually make visible to you through this virtual session something that is difficult to see unless you have this wonderful model in your hand and you're sitting at a table with a couple of students and you're actually pointing and touching and sharing this. So I hope that if nothing else, what this will do to you is make you want to have one of these things in your hands. And yes, you can buy these things from 3D&D, but more importantly, in these days of low-cost 3D printers, you can have your kids making these models as classroom materials that you will acquire a collection of these things that your kids build over a number of years. And suddenly you have a whole collection of physical models of important proteins you want to talk to your kids about. But I don't know if you can see it, but there is a single chain of water molecules that comes down through the pore, the channel that's formed by this 7 helix bundle. All right, so water goes through in a single file way. It's highly selective for water. It doesn't let anything else go through, and it doesn't even let protons through. Now, you know, a lot of times a membrane, think of mitochondria, the inner membrane of a mitochondria. We set up a proton gradient across that membrane, right? We use that to drive the ATP synthase that makes ATP. So we certainly don't want protons to be able to make its way through a membrane. And we've got a water channel, and it was well known by biophysicists that protons like to associate themselves with water molecules. So people worried about, you know, how can we transport water specifically across the membrane without also letting some protons leak through? So this protein has evolved to solve that problem. So I'm going to take another model here. How am I doing on time? Am I way over? Yeah.
We have less than 5 minutes.
Okay. Well, all right.
We'll
so-
stay on for questions afterwards.
Right. So this model is a little larger, and you see it has a simpler color scheme. It has a blue half and it has a green half. And if I could have that model that Augrey, the 1992 model that Pierre Augrey came up with back for just a second, I want you to look at the shaded area, and I want you to see that the first 3 alpha helices are sort of symmetrically related to the last 3. You see that twofold symmetry there? Well, that's what's reflected in this blue half and this green half of the protein. So if I separate these, and if I were to lay these down, and my overhead camera is not working, so I have to do it this way. But what I'm going to try to convince you of is that half helix that we talked about, the green one comes down right here, and there is an asparagine residue at the very end of this helix. So now I'm going to pick up the other half, which of course has the blue half helix coming up from the bottom. And guess what it ends in? It ends in an asparagine. And in fact, all aquaporins, which there are 13 human isotypes of this protein, 13 different kinds or variants of aquaporin in human tissues. And all of them will have that invariant asparagine just at the tip of these 2 half helices. And what happens is that this is how we ensure that protons don't leak out across, don't hitch a ride on the water that goes through a channel like this. And the best way that maybe I can explain that is just with my hand. So I want you to imagine a chain of water molecules lined up here. The protein is right here. The water molecules are going from the outside through to the inside. And right in the middle, we have a sort of a pinch point where we have one helix, half helix coming up from the bottom with an asparagine, another half helix from the top, asparagine. So you have 2 asparagines right here with sort of the nitrogens pointing into the channel. The water is coming down here. And, you know, a water molecule, you know, has 2 hydrogens, right? And when that water molecule starts coming down, those hydrogens are always pointing up until they get right here, halfway through the aquaporin, where you have 2 asparagines. And at that point, the oxygen of the water rolls over. So it interacts first with the first asparagine, and then with the second asparagine. And as a consequence of rolling over from the 2 asparagines in the middle, then all of those water molecules continue to filter through the pore with the hydrogens pointing down. So it's up, 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 roll over, down, 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 down. Now, how do we know that? We know that because there will be a link to a molecular dynamics group at University of Illinois, Champaign-Urbana. So there's a really high-powered group there that does molecular simulations. So they take the structure of the aquaporin and the side chains that line that channel, and they put a bunch of water out here, and they put a bunch of water in here. And this is just osmotically sensitive. These are not gated channels anyway. They're just open. And the water will decide, which way it wants to flow based on the osmolarity of the solution inside and outside. So they can simulate this computationally. And they will simulate maybe a microsecond of real time of these molecules jostling around. And of course, they assign various energetics to the hydrogen bonding that occurs between the water molecules and the side chains that line the channel and so forth. So you can actually see this happening. Now, you don't really see it happening, but they've modeled what would happen with molecular dynamics. So when that Nobel Prize was announced in 2003, this group in Illinois had up on their website within a few hours of the announcement this molecular dynamics simulation, which shows those hydrogens pointing up, going over, going down. So this was, you'll have a chance to link to that, an animation, a little movie that shows that, and it's really a cool thing to show your students. So there are more things we can say about aquaporin. Those two half helices. The alpha helix has actually a dipole moment. We don't have time to tell you why that is, but come to the course and you'll make an alpha helix from individual amino acids. And you'll see that the nitrogen, the internal end of an alpha helix, has sort of a positive charge, not a full charge, but sort of positively charged, a positive dipole. And these two half helices that point into the middle, it's the N-terminal end of each helix that is pointing into the middle. So it's like you're focusing a macro dipole right there at that selectivity filter in the middle of an aquaporin. So that's another interesting feature of how the protein has evolved to allow water molecules, but only water molecules, very small water molecules, to move through this in a very, very particular way. All right, I don't know if any of that made sense. I'll say one more thing, and then maybe we can entertain some questions here. I was amazed 3 or 4 years ago. There was another paper that was published with a high-resolution structure of aquaporin. And when I say high resolution, that's what was so amazing. This was a sub-angstrom resolution, meaning the resolution was, you could see features that were less than 1 angstrom. It was a 0.88 angstrom structure. And I didn't think it was possible for people to determine a structure with that much detail. But at that level of detail, they could actually see the individual water molecules very clearly defined in this channel. And the only thing I will say is that as a result of that work, then, all models are wrong. And what we know that's wrong about this model that shows a single chain of water molecules is that as a result of that high resolution structure, they think that every other water molecule is missing. What am I trying to say? You don't have water. You have water space, water space, water space. So these things kind of move through the channel in pairs with an empty space between two adjacent water molecules. And actually, that's another feature that when we talk about the potassium channel a little bit later, you'll see that a very similar thing happens with potassium. Potassium goes through in sort of pairs with space in between. All right, so are there any questions? I don't know if that made sense. It's a good story. Your kids will enjoy the story because Peter was not Peter is a Norwegian from Minnesota, And he finished high school going to night school. I have something to do with the D that he got in chemistry during his junior year. So he became a world traveler. I think he was a hippie. Maybe he was a hippie Norwegian from Minnesota.
And then with that, he was at Augsburg University, where Jim is doing some adjunct teaching.
Yes, Jim, I didn't make that connection until Heather mentioned that. But yeah, I wonder if Peter Agre is a legend around Augsburg, because you get a D in chemistry in your junior year, you finish out your high school degree in night school, and then, by the way, you want to go to medical school. Well, that kind of a high school experience doesn't set you up very well to get into medical school, which is pretty competitive. So Peter went to his dad's college, which happened to be Augsburg, which is by all accounts a very fine school. Everyone who went there, all the pre-meds who went there, got into the University of Minnesota Medical School. So they've got to be doing something right there. So yeah, that's an interesting connection with Jim.
Okay, so I want to wrap up the formal part of our session now. We will stay on for questions and answers, so hold on for just a minute. But thank you guys for a great session tonight. Reminder to receive a certificate of participation, click on the link that Diane's posting. Jim used the water kit. We used the sodium chloride lattice and water student modeling pack today. Tim used, well, didn't quite see our aquaporin models, but we have a couple of aquaporin models available for sale.
Yeah.
Use the discount code webinar20 to get 20% off most of our products. I hope that you'll join us again next week when Fran Grant and Mark Arnhold from Wisconsin present transcription and translation, and with editing beta globin. So please encourage your colleagues to register and join us. When you do leave the webinar, you will encounter a short survey. Please complete that. We really do appreciate your feedback. And thanks again for joining us. We will go ahead and do questions and answers now. So raise your hands if you'd like to be promoted to the stage, or go ahead and post some more things in the question and answer, and we'll try to get through all of that. And please stay on just to say hello. We did have one question,
Okay.
and they were asking about do you know when they actually crystallized aquaporin, and did they have to do anything special to get that structure?
Oh, I hate questions like this because the answer is no. Have you ever stood in front of your class without having properly prepared for whatever the topic is of the day? If you have, you know how I feel right now. I can tell you the paper, first author in the paper is Murata. And there will be, I guess there will be now that you've asked the question, there will be the PDF of that paper will be part of the resources that you will find associated with this topic in this webinar series. So I don't know for sure, but I'm guessing maybe 4 or 5, probably maybe 3 or 4 years after 1992, so in the mid to late 90s. And I only say that because that would have given the Nobel Committee time then to know that, oh, this is a nice story. They discovered the protein. They showed it was a water channel. They solved its structure. The structure explains the function of the protein, how it can transport water without letting other things like protons through. So, you have to let these things percolate long enough so that so that the Nobel Committee feels as though it's worthy of a Nobel Prize.
Thank you. Sorry to put you on the spot. Edward, would you like to come on and ask a question?
Okay, I see the word Edward McGee, but it looks like Edward is still muted.
It may be that Edward had his hand raised before we invited people to the stage, so
Okay.
I invited you to the stage.
Let's put Edward on the
My
spot.
apologies.
So We have a question about why would a red blood cell need to take in water quickly?
Yeah, now I already told you I don't know the answer to that. It's not apparent to me, but actually I think I do know the answer. You know, red blood cells have this very unusual biconcave shape. So maybe the question is, why do they have this biconcave shape? And it's because the diameter of capillaries in the finest part of a capillary bed, the diameter of that vessel through which a blood cell is flowing is actually smaller than the diameter of a nice plump biconcave red blood cell. So the shape of the red blood cell allows it to squeeze through these little capillary beds. It's actually smaller than the red blood cell itself. And that doesn't quite answer why water flow in and out is important, but it might. I don't know. The answer doesn't have to be right. It just has to be convincing to your students, right? So I could imagine if you were a red blood cell that was squeezing through, a tight door, osmotic pressure on the inside might need to be relieved a little bit as you went through, and then once you got through, you might want to water back in. I don't know. That's a total guess. I don't know. But it is obvious that there are other tissues, and the most notable one is, of course, the kidneys, where we filter urine and we have to reabsorb lots of water. Otherwise, we would be dehydrated. We'd be drinking gallons and gallons of water every day if we weren't able to resorb water across that membrane. And the loop of Henle. It's the only thing I know about a kidney is that there's a loop of Henle, and that's where you find all of these water channels to do the water reabsorption. It's also important in the eye. Again, I don't quite know why, but there's a lot of water transport going on in the eye.
Great. And Kathy and Mary, I know you had posted some questions in the question and answer. Did we get those answered? Or if not, can you expand on those a little bit more? I think I missed the first half of those. Or come on. And I believe that Jim is available to answer questions as well. So raise your hand. And we have another one that was posted here. How far back in evolution can aquaporins be found? Sponges?
All right, well, I'll take the last one because I can quickly claim I don't know, and I'll let Jim answer those hard ones up above. I don't know, but my guess would be way, way, way back, because all of life forms on Earth are aqueous-based life forms, right? So from the very simplest cells, you had to have a membrane that separated the outside from the inside. And because we are aqueous-based life forms, then water makes up 70% of the mass of cells. So you've got lots of water inside, you've got lots of water outside, of course, that's where life evolved. And then you've got this issue of transporting other things across those membranes. So if you transport a bunch of ions on one side of a membrane, then the water is going to follow, right? Water always moves to try to equalize the concentration of a solute on either side of a membrane. And that must have been one of the very first sort of fundamental things that life had to deal with is how do we move water around in response to increasing concentrations of proteins, of molecules, of osmolarity across the membrane. So I would guess that these are ancient proteins. I don't think you find a life form that probably doesn't have some sort of an aquaporin in it. You can ask about the chicken and the egg. You couldn't have life before you had an aquaporin. So, all right.
Okay. So, and thanks to Dan. So, hello, Dan. He was recommending coming to the summer course. He's been to several workshops. So good to see you, Dan.
Who is Dan?
Dan
can't see
Finsky.
Dan. Oh, sure.
Mm-hmm.
Okay.
And so do you want to do the 6 water ring or should I?
You do one and I'll do one, and then we'll do ice together, okay?
And I have one pulled up too.
Oh, okay, Jim, do you want to do ice? Or is that what you meant, Heather?
Yeah, I can definitely do the 6 model or the 6 molecule ring. So here I have the six molecule ring, and one way you can tell you've done it correctly is you have these hydrogens kind of pointing up, or these hydrogens pointing down, and you can see there's all 6 are right there. So there is your 6 molecule ring, and I know we can talk about cubic ice and all that, but that is to answer that question directly.
Yeah. So
There is this dual
we
ring.
can do this. This is one of my favorite activities with a water kit, and that is, everybody knows that ice floats on top of lakes, or the ice cube in a glass of ice water always floats at the top. So why does ice float? Most liquids, as you cool them down, they get denser and denser. So you would expect that ice would, as the water cools down to freezing, it ought to sink to the bottom of the lake. And when it finally gets to 32 degrees, it would solidify, and you'd have ice on the bottom, and you wouldn't be here because life could not have evolved in water under those conditions. So water is very unusual in that when it gets down to about 2 or 3 degrees above freezing, so maybe it gets down to about 32 degrees and it's getting more and more dense, but then something happens when you go from 34 to 32 degrees. And what happens is this: I've got 2 rings of 6 here. And if you watch carefully, I'm going to take this one and I'm just going to rotate it in my hand by 180 degrees. And again, you're not going to be able to see this in detail, but now I've got 2 identical 6 molecule rings, one rotated relative to the other. And listen, the most important thing is listen to the click. Did you hear that click? That is the formation of a hexagonal ice lattice. And the point is, it's an expanded lattice, open lattice, and it's made up of 12 molecules of water, right? But I can stick my finger right in the middle of this, and in fact, I can take a 13th water molecule, and there's plenty of room in the middle of that for a 13th water molecule. But in fact, when water freezes into this open lattice, there is no water inside there. So the density of this ice lattice is less than the density of liquid water, where that 13th molecule would be in there. And I'm going to take this beautiful open lattice, and then I just start moving all of these around. This is liquid water, where those hydrogen bonds are forming and breaking and reforming. Okay, so the density of liquid water is greater than the density of ice, and therefore ice floats to the top of the lake. So I think this is really one of the most amazing things that you can do with a water kit. It's not easy to make these ice cubes, but once you've done it for 20 or 30 years, it becomes routine. So there are instructions on the website, I'm sure, to do that. Yeah, Jim, I think Jim's doing it. Is that right? Yeah.
Yeah, so you can see the one side has the oxygens kind of pointing up, the other side has the hydrogens. And if you take 2 of those together, you can hear that whoa, and then you drop it and it all falls apart. But then you can see it does create that,
And
that
then,
lattice.
and then the last thing you can do, and this is what we're doing with the grandkids recently, is what you know about snowflakes and the symmetry of snowflakes. All snowflakes have 6-sided symmetry. So 6 hexagonal ice. So you start with rings of 6, right? So the reason that all snowflakes have 6-sided symmetry has got to be related to the fact that you start with these hexagonal rings. So if I had the ice lattice here again, the second layer, you could count 6 here, you could count 6 on the side, 6 in the back, everything is 6, 6, 6, 6. So when these hexagonal ice cubes start coming together way up in the clouds under the right conditions of temperature and pressure, where you form snow, then you form these beautifully symmetrical snowflakes with 6-sided symmetry. That actually blows my mind. And if you think too hard about how that happens, I don't believe it can happen. And yet I've seen people who go out in the cold of Minnesota and capture snowflakes onto a frozen microscope slide. And they photograph these and they publish books then that show these beautiful snowflakes. And whatever strange structure, builds off of the snowflake over here, the same structure is building off over here on the other side of the snowflake. Now, you explain to me the laws of physics that are ensuring that symmetrical building out of a snowflake from a common core. That's just amazing.
I can see that we're going to have to create a video on how to make the ice cubes and cover some of these other things. But we do have a question in a different tract,
Okay.
going back to the aquaporin. And let me read it. Are there any notable features on the extracellular side of the 7 transmembrane proteins as compared to the cytoplasmic side?
Yeah.
Other than hydrophobicity? Yeah.
Again, I will preface my answer by saying I'm not an expert in any of the things I talk about, and therefore you have to take everything you hear from me with a grain of salt. So my answer is no. I'm not aware, I haven't read anything about any notable differences between the outside and the inside, the exposed surfaces of an aquaporin. And that kind of makes sense to me because remember I said that the water flows in both directions. Many of these channels are gated, and therefore you'll find on one side or the other, you'll find another protein domain that often undergoes a conformational change to sort of plug the pore. And then it only opens in response to a voltage difference. So when we talk about the potassium channel, these are voltage-gated potassium channels. So there, the intracellular surface where you find the gates is very, very different from the extracellular surface. But in the case of aquaporins, because you have this bidirectional flow of water in either direction, I'm not aware of any notable sort of structure function reasons for why the outside surface is different than the inside.
There's one question left in the question and answer section about showing the sodium and chlorine clumped in the water and open the hand and the sodium chloride was reforming. So I don't exactly know the context there. But I think if you look at when you are if you just kind of crumple these in your hand, you'll see that it's quite hard to get that sodium and chlorine to come apart, which really then you can talk about temperature of water. So the more energy that there is in those water molecules, there's a minimum amount of water and energy required to kind of to break that bond. But when we want something to dissolve, how do we stir it? We're adding that extra energy to help facilitate that connection right there.
I've never tried to teach this to high school kids, but I think it's true that hot water will dissolve more salt than cold water, right? So that's kind of a nice experiment that kids could do that would illustrate that point.